Good evening, Divya. Okay, I'm sharing my screen. Okay, so today we are starting the final and most important part of this course, that is the project. So, till now, you have learned concepts, you have learned tools, you have learned basic bioinformatic ideas. But in real bioinformatics, we don't just learn tools, we apply them to answer biological questions. So, today I'm going to explain one complete bioinformatics pipeline project, and my role here is not to do the project for you, but to guide you on how researchers think and work. So I have designed this project specially for you, and the thinking behind it is exactly like the same as real research. So our project title is In Silico Identification and Functional Characterization of a Target Gene or Protein using Sequence Analysis and Homology Based Annotation. I know this title looks big, but don't worry, let me simplify it for you. So here, in silico. In silico simply means that we are doing everything on computer. So in silico clearly shows that this is a computational project. Then we have identification. So identification means finding out what the sequence is similar to. And then we have functional characterization. So it means predicting what this gene or protein might do. Then we have the target gene or protein. So here I have mentioned both gene and protein. So you can choose either gene or you can choose protein. It totally depends on you. Okay. Then we have the sequence analysis. So it means checking the length, checking the GC content and basic properties. And then we have homology based annotation. So homology based annotation means using similarity with known sequences to predict your target gene or your target protein. So in simple words, this project is about taking a sequence and using bioinformatics tool to understand what it might be and what it might do. So this project focuses on analyzing a biological sequence and predicting its function using computational sequence analysis and similarity search. Now we have a core research question. So every research project starts with one question, and our core research question is, given an unknown or hypothetical biological sequence, how can we computationally analyze it to predict its quality, to predict its similarity, and to predict its biological function? So this one question derives this entire project. And here we are not randomly using tools. Every step in this project exists to answer this particular question. Now, the important concept that this is a single continuous pipeline. So, this concept is very important, and it means that each step depends on the previous step and we cannot skip any of these steps. Okay, so we move logically just like the real research. So, in bioinformatics, pipelines are more important than individual tools. So here we have the step 1 of this project. So firstly, we need a biological sequence. Okay, and this biological sequence can be a gene sequence. You can either choose a protein sequence Or if you are interested in viruses, then you can choose a viral gene or any hypothetical proteins or disease related protein as well. Okay, but notice something very important here, that we are not starting with the tools. We are starting with a biological question and a sequence of interest. So this is exactly how research begins. Now, once we have a sequence, we don't blindly trust it. So in step 2, we perform basic sequence analysis. So here we will use the Biopython to calculate things like the sequence length, its GC content, or if your sequence of interest is a protein sequence, then you will calculate the amino acid composition for proteins. So here the purpose of this step is not calculation, the purpose is validation. So before moving forward, researchers always ask, like, does this sequence look biologically reasonable? And then comes the third step. So here third step is extremely important because now here you apply biological criteria. For example, removing very short sequences or removing low quality sequences. So this step teaches you something very valuable, and that is decision making. So in real research, scientists constantly decide that is this data good enough to move forward. So this step stimulates that exact thinking. Then we have the step number 4. Now we reach the heart of the project, and here we perform homology search using BLAST. So here you will identify closest matching sequences, you will identify the conserved regions, you will identify the evolutionary hints. But remember this line, that homology is the foundation of functional prediction. So what we are doing here, we are using BLAST, and we have already learned that what BLAST does. So we find the similar sequences to identify what our sequence of interest is, what is it, what is its function. Okay, then we have the step 5. So step 5 is about the functional annotation. So using the top blast hits, we have already learned that why do we use always the top hits of the blast. So using the top blast hits, you will retrieve annotations from the UniProt. You have learned UniProt as well. So here in this step, you will predict what the protein or gene does, its biological role, and the organism it is associated with. So in bioinformatics, function is inferred, it is not experimentally proven. Okay, so this is a very important concept. Then finally, we reach the step 6, that is biological interpretation. So here you will write that what does your sequence likely do, what and why do you think so, and what evidence supports your claim. So this step is what that transform your work from just an assignment into a research style project. Now you will also maintain a proper project structure. So it's not only about the code, it's also about the organized files, how you organize your data, how you organize your outputs, and how you organize your explanations. So this is how the professional works. So by the end of this project, each one of you should be able to confidently say that I analyzed an unknown biological sequence, I validated its quality, I identified homologous sequences, and I predicted its biological function using the computational methods. And this is a real bioinformatics logic. So here I will guide you in this project. I will explain you the logic and I will help you when you are stuck, but I will not do this project for you. Okay. So the goal of this project is not about its completion. The goal here is learning how to think like a bioinformatician. So by the end of this project, I want you to be confident enough to say that you can take any biological sequence and you can analyze it logically and you can explain why you are using each tool and what the results mean. So this project is not about writing the perfect code. It's about developing the scientific thinking, the problem solving skills, and understanding how the real bioinformatics research is carried out. Okay, so focus on learning. You can ask questions when you are confused and try to think like a researcher. So this is the real objective of this project. And also for your reference, I have added one more slide here, how you can choose your sequence of interest. So there are four options. You can take a look at it when you will start the project. So from these options you can choose your sequence of interest. So if you have any questions, you can ask. Gargi, in any format that you feel comfortable. If you feel like you have to make a PPT, you can do that. If you feel like you are more comfortable with MS Word, you can do that. So the thing here is you will write the code and you will also explain why you are writing the code, how you are doing this project. So that's why I made this slide, this one, that how you are organizing your project. Okay, so you can write in points, but also one more thing, that you will be given 10 days to do this project. So by 5th of February you should submit your project. I will also explain you how to submit in on GitHub, how to upload it on GitHub. So if you students are interested in giving the presentation, so what I will do, I will divide you into groups. Okay, and each group will consist of 6 students. So if you are interested in giving the presentation, so I will send the poll in the WhatsApp group. Okay, how many students are interested in giving the presentation? So those students, I will divide those students into groups and you will make a group. Okay, wait a minute. Pranjali, that's the thing that you will research about. Okay, Pranjali, you will explain this in your project. Gargi, are we clear with the format? You can make any format, you can do it in whatever way you are comfortable. Yes, David, you can continue it. Yes, Virasri, that's why we are doing this project. So the main thing is, if you do give the presentation, it will build your confidence. It will help you understand your own work, how you started from the first step and then how you analyze your sequence. The thing is, you have to take an unknown biological sequence. You are not allowed to look upon it what's its function. Okay, you have to take an unknown sequence, because that's the whole thing about this project, how you can analyze any biological sequence using the tools that you have learned. Okay, so in this project, what we have learned during this entire classes, we learned about Biopython, we learned about BLAST. So that's whole I have created in this single continuous pipeline. Okay, that's why it's a single continuous pipeline. So these are the steps that you are going to follow. Okay, so you will be given 10 days to do this project, and I will send a poll in the group if you are interested to give a presentation. So I will divide you students in a group, and each group will consist of 6 students, and each single student of a group is required to be give the presentation. Okay, so each member of the group is required to be involved in the presentation. Vidasri, that's why I told you that I have added a extra file here for your reference. These are the 4 options from where you can retrieve your unknown sequence. Okay, so take a look at it, research about it, try it, go to Uniprot, go to NCBI, or if you are interested in viruses, I have added this option as well. So these are the 4 options where you can retrieve your unknown biological sequence. Or if you have your own sequence of interest, you can work on it as well. So it totally depends on you if you want to work in a group or you want to work independently. No, Sudeshna, no, the presentation is not important. I mean, the presentation is not mandatory, but it's important for your own. Okay, it will give you a benefit to build your confidence, because when you will give interviews, the interviewer will ask you about this project. what is the project about, what did you do in this project, what are the tools that you used in this project. So if you give the presentation, you are preparing yourself to give those interviews very well. Okay. Okay, Tisha, single continuous pipeline, it means that all the steps depend on each other. Okay, so the first here we have the first step, that you will retrieve your sequence of interest. Okay, then you will perform the sequence quality analysis. So if you don't have your sequence of interest, on which are you going to perform the quality analysis? Okay, so each step depends on the previous step. So that's why it is called a single continuous pipeline, and that's why pipelines are very important. Yes, with Asri, you can select any sequence of interest. Lakshana, I will divide the group. I will tell you how, I mean, I don't currently know everyone of your name because not every student attends the class. So that's why I will just add a poll in the group that how many students are interested to give the presentation. Then I will make a group and then I will let you know that you are in this group and these are your group mates. Okay, these are the people that you are going to work with. So you can chat with them how you're going to do this project. Okay, Muskan Yadav, as I said that you will be given 10 days to complete this project. So on the 5th of February, you have to submit your project, and from 6th to 10 of Feb, okay, 6, 7, 8, 9, 10. So I will just text in the group that today which group is going to present their presentation. Okay, so on 6th onwards, 6th Feb, you will be giving the presentation. Shraddha, you will be presenting your project. Okay, this is the project that you are going to work on. So it totally depends on you if you want to work independently or you want to work in a group. Muhammad Ali, that's what we learned in this all classes. We learned how to use Biopython. Okay, as it's a research project, so you have to do your own research as well. Okay, you can always text whenever you are stuck, but you are going to do this project on your own. Yes, Aditya, as I said, it's totally depend on you if you want to work individually or you want to work in a group. But if you want to share a presentation, you have to work in a group then. Abhay Patel, if you want to present, if you want to work in a group and you want to give a presentation, then by the 5th of February, you have to submit your project.